So I have the unique, distinct, singular, where's Jonathan? Find me another word, pleasure of introducing a legend and a rarity in today's world of medicine. And I'm going to ask what we call Danon disease to be perhaps repronounced properly by the doctor himself when he comes on stage. And I hope this can be a moment where we learn together how to really pronounce what we call Danon disease. Getting to that in a second. We had dinner last night and we talked about this. So Dr. Moris Danon, he identified Danon disease in 1981. And since then, awareness has grown in leaps and bounds in recent years. He's a professor at Tulane University as well as Louisiana State University. And he's trained as a clinical neurologist specializing in neuromuscular diseases, but as he likes to call himself, he's a morphological neurologist. He's very much focused on morphology of cells and tissue, and that has a lot to do with the genesis of Danon disease and all of his work over the many decades. And a fellow New Yorker as well. So I would ask everybody to rise in great respect for Dr. Moris Danon. So, first questions first. How do you pronounce your last name?
Well, originally it was Danon, and when I arrived in the United States, I met a rather large blonde nurse in Queens General Hospital who said it's Danon and it's stuck. So that's how the name thing goes about. Is this thing working?
Put it up like this.
Yeah. Is it working? Yeah, okay. Yeah, all right.
Would you like us from now on to pronounce this disease Danon?
No.
Oh, okay. Okay.
I never forgotten the big blonde nurse.
So you have a captive audience here. Everyone is fascinated with you, here in the room as well as online. Tell us the story of how this disease came to be named after you.
Well, let me tell you about my background a little bit. I'm a card-carrying neurologist, and my hobby is nerve and muscle pathology, immunocytochemistry, histochemistry, and electron microscopy. And I was a young attending, just finished my fellowship in nerve and muscle pathology at Montreal Neurological Institute. And I set up my lab in the University of Illinois of Chicago for muscle and nerve pathology. And this was my third muscle biopsy as a young guy. And at first I didn't know what I was looking at because it was such a rare findings. And then we have the slides to show you afterwards how these things look like. And then, of course, I didn't realize it was a new disease, and that's how we published it.
So before we get to your highly awaited presentation and slides.
I'm sorry?
Before we get to the slides themselves and the presentation, what was your experience?
Clinically?
Right. Before we get there, I have a couple of more questions for you.
All right, sure.
So how did you get excited about the field of medicine to begin with, Dr. Danon?
Well, when I was a young kid, a very young and good-looking internist took care of me, and I said I want to be like him, and I did.
Where was your training?
I went to medical school at the University of Istanbul. It's sort of an attractive city. And then I trained in neurology at Northwestern Hospital and St. Vincent's Hospital in New York, which does not exist any longer. Now it's condominiums. And after that, I went to Montreal Neurological Institute, and I trained my first year in neurophysiology of muscle. And my second 18 months, I did nerve and muscle pathology, light microscopy and electron microscopy. And that's about my training. And I did pass my neurology boards.
Tell us about your experience at Queens General Hospital.
Well, that was a tough place. And as an intern, we walk in with our white coat and come out in blood. We had a lot of Jamaica, Queens, and there were a lot of shootings and stabbings. And in my OB rotation, I delivered 75 kids in 45 days. And I was doing episiotomies with my eyes closed.
How do you feel today, looking around this room and knowing that your fascination and love for medicine and morphological neurology has led to the diagnosis of so many patients and families who were uncertain, as we heard, about what was going on. Now we know what these patients and family are dealing with, and potentially we have a path forward toward cures. How do you feel sitting here in this audience today? What does this moment mean to you?
I think right now it's overwhelming with this crowd, but other than that, neurology is sort of an exciting field. It's a thinking man's specialty. And we did deal with rare diseases all the time, especially when you're in the field of neuromuscular diseases. And it's always good after the unknown and try to find out new things.
You're a neurologist. Right now we know Danon disease to be primarily a disease of the heart. How is it that a neurologist discovered this disease? Why?
Most scientific neurologists also have subspecialty, whether it's nerve and muscle disease or immunology or genetics, and that's what certain neurologists are different than clinical scientist types. And they consider one of them.
What does the word Danone mean in Turkish? You taught me this last night.
What?
You taught me the meaning of the word Danone.
Yeah. Oh yeah. That's the famous yogurt, and and I think that was so catchy that the Japanese named the disease after me.
Dr. Danon, if you have one message for the patients and families in this room, as well as for the scientists working very hard day and night to move toward cures, what is your message for us?
My main message is obviously don't despair. We're really working very hard on it, and hopefully in a lot of rare diseases, there is a lot of advancement, whether it's this year or in 10 years. And believe me, there are a lot of people in the basements of their labs doing a lot of strange work to help at the end patients who have rare diseases.
I'll ask one last question because I know where you're headed this evening. What is your favorite restaurant in New York City?
The what?
Your favorite restaurant in New York City?
Oh, Le Bernardin.
So I'm going to open this up for anyone who has questions for Dr. Danon in the audience, and then we're going to ask him to present some of his work to us. We're just warming up. Don't be shy. Paul.
Good to see a fellow LSU. I'm from Louisiana as well. I know David's back there. So go Tigers, all right? Hopefully win the national championship next year. So I wanted to ask, what was your experience following the patients who you initially sort of, you saw their initial biopsies? What was your experience following them?
Your experience with patients following their initial biopsy? Yeah.
Well, not much. I didn't follow these patients, and we didn't even know we were dealing with a new disease at that time. And if you look at the literature, there's quite a bit of information about the natural history of the disease, and which the main problem is not the muscle disease, really is the cardiac involvement. And that's what they need to treat first, because of most of those patients came about because of their heart disease, and then they were discovered to have primary muscle disease.
Thank you.
So I think I heard you say you found out that in Japan, the disease was named after you. How did you find out there was a disease named after you, and what was your reaction to that?
In Japan, you found out that the disease was named after you. How did you find out? What was the moment you found out, and what was your reaction?
Well, I was in a symposium at NIH for Pompeis disease, acid alpha-glucosidase deficiency, and there was a big Japanese group who were interested in Pompeis disease, and at that time there was some advancements in Pompeis disease with the enzyme replacement, and that was the focus. And then they asked me to present my case of this disease, which feels funny to call your name a disease. And so about 3 Japanese physicians came around and asked me to take a picture. And I said, all right, you know, and then all of a sudden 20 people showed up for the picture. And I didn't realize why. And I found out in the 1980s that the disease was named after me.
Show us this paper. This is the original manuscript, the original publication.
This is the original publications.
So that's the publication where the disease was first characterized clinically. And in 2000, this is the link between Danon and LAMP2 in Nature. Really remarkable, really remarkable. So I will hand the podium to you, Dr. Danon, to present some of your work, and then we're going to invite our esteemed panel of fellow cardiologists and internists to join us on the panel. So I'll hand it over to you. And then...
Well, this is the first case diagnosed with this disease when I was a young attending in neurology and running the nerve and muscle pathology lab. And this was my 3rd biopsy in my career. And it was an unusual one, obviously. And this was a 16-year-old boy who walked late around age 2.5. By 4 years old, he couldn't climb stairs. And by 10 years old, he had a cardiac arrest, and he needed to be resuscitated and defibrillated. And then he was diagnosed with catheterization to have a severe cardiomyopathy. While he was admitted in cardiology, he was referred to me because they just noticed some upper extremity weakness, especially with the arms, and they said it might be also a muscle disease, and the 2 of them might be connected. And at that point, they did a serum CPK test, which showed high serum CK, suggestive of muscle breakage. I did an electromyographic study on him myself, and showed a rip-roaring myopathy with spontaneous discharges, which was fairly typical for actually for Pompeis disease. And then we biopsied him. And these are the biopsies of this first child. And as you can see, some muscle fibers have holes in them. In our parlance, it's called vacuoles. And this is in higher magnification. As you can see, there are tremendous holes and just some muscle tissue left in the middle of the muscle fiber. And this is even higher magnification, showing the major holes in this muscle. This is like a little puzzle, actually. As we're going to go through stains, you will see how we came to the bottom of this illness. And as you can see, the muscle fiber on the very right, there's a big hole and junk in it. We don't know what the junk is at this point. These are regular H&E and trichrome stains. And as you can see, in this fiber, you can see a lot of red dots. And that's suggestive usually of storage in muscle. And as you can see in higher magnification, this hole in the muscle and with junk in it. This is a myofibrillary ATPase stain showing histochemical type 1 and type 2 fibers. And you can see the holes are in both muscle types. And this is a PAS stain, which is positive for sugars. And as you can see, the fiber on the very right and the bottom has red blotches in them. These are PAS positive materials suggestive of some sugars. And as you can see, this in higher magnification, this fiber on the very right shows a tremendous amount of very red accumulation, which is sugars. And when we use a diastase stain on a PAS stained tissue, it clears up only glycogen. So this slide shows that the accumulations which were digested by diastase was glycogen. So we were dealing with a glycogen storage disease. It's like a little puzzle, actually. And then this is an acid phosphatase stain, which is a marker for lysosomes. So the same holes that are PAS positive and diastase negative also have this very dark black blotches in them, suggestive we're dealing with the lysosomal glycogen storage disease. And as in higher magnification, you can see the acid phosphatase accumulation in middle of the fibers, in two fibers, suggestive there is a lysosomal glycogen storage. And there is only one disease that causes that, and this is acid maltase deficiency, also called Pompeis disease. And this is a fat stain showing little fat droplets in normal muscle. And the reason we did the stain was make sure this was not a lipid storage myopathy. So that ruled out with various histochemical stains where we were going with this. And then the electron microscopy, la pièce de résistance, as they say in French. And this is, you can see the muscle fibers here practically destroyed, and there are a lot of dots all over the place. Those dots are glycogen. And some of them are in membrane-bound spaces. These membrane-bound spaces are lysosomes. That means there is a problem with the lysosome, which is found in acid maltase. When you have acid maltase deficiency, you have a lysosomal glycogen storage disease. And what happens is the lysosomes cannot digest the glycogen. They burst and they destroy all the muscle tissue. And this was what we found in this child. And this is even a better presentation. And LY stands for lysosomes. You can see all the membrane-bound structures diagnostic for Pompeis disease. And this is even in higher magnification, all the way down, we can see the lysosomal glycogen storage. At that point, oh, this is my last slide. This is a very interesting slide, and it's a slide representative of Pompeis disease, except this is not in humans. This is in genetically engineered knockout mice with Pompeis disease, which is identical to that. But this is mice. So if you take this picture, show a human picture and a mouse picture, you cannot tell the difference whether it's human or mouse. So I thought I'd throw in for fun. This is work I did. So then I sent the muscle tissue to the biochemist at University of Illinois, a man called Louis Schlissefeld. And he's really the unsung hero of this disease, and he's in the first paper. And he came to me, he said, well, he said, the glycogen content is increased, but the acid maltase is normal. So all of a sudden we said, so it's not Pompeis disease. So I said, repeat this darn thing because it's impossible. This is Pompeis disease. It's textbook. So he repeated the whole thing, then he repeated all the enzymes of the glycogen metabolism. Again, high glycogen content, but no abnormalities in all the glycogen circuit enzymes. So I called Dr. DeMauro at Columbia University, and I said I had this case, and it's typical Pompeis disease, but it's increased glycogen, but normal acid maltase. So I sent the muscles to him, and he reproduced all the studies and indeed found exactly the same thing. At that time, a second case came in from University of Alabama, Dr. Shin Oh. The same thing with this child with mild mental retardation. They all went to special schools. Then all the children turned out to have the same triad, and cardiomyopathy and muscle disease. So it turned out to be also a clinical triad of this disease, in addition to the muscle pathology. And then in the year 2000, in the Journal of Nature, Dr. Nishino from Tokyo Brain Institute and Dr. DeMauro and Michi Hirano from Columbia University nailed down the lysosomal defect, which was secondary to LAMP2 deficiency, structural protein, which resides in the lysosomes of muscle. So that's the story of this disease, which is really a clinical triad with specific pathological abnormalities and a specific lysosomal defect, which you people are working to try to correct it. And I hope you'll be very successful.
Amazing, amazing story. And I think a lot of us here, we're glued to some of these slides, and we want to learn from you how to properly fix some of these slides so we can have better and better EM and immunohistochemistry and others. Eric and I were talking about it this morning. We've got to get the pH right is the key, right? So we're now going to be joined by some of the most accomplished physicians and physician scientists on this planet. Starting on the far end is Dr. Eric Adler. Eric is the reason that all of us know Danon disease and the work of Dr. Danon. He produced the first preclinical gene therapy evidence that we could have a viable treatment for patients with Danon disease. These were beautiful slides. Similarly, when we saw Eric's first slide several years ago demonstrating a complete eradication of these vacuoles in a preclinical model, we were stunned and moved to action. And we're so happy to have you as part of our extended group. And he's also my Sunday workout buddy, by the way. So. So actually, I'll move next to Dr. Matt Taylor, who, like Dr. Danon, has been seminal in the description and diagnoses of Danon disease patients. He published the first natural history of Danon disease over a decade ago now. He's a research scientist and a physician at the University of Colorado, and has become a friend over the years that we've gotten to know one another. So welcome to Matt Taylor. Thank you, Dr. Matt Taylor. Dr. Barry Greenberg, one of the coolest people on this planet, first of all. You should hear about his stories. It's a role model for many of us in this room who are not so cool. And he has several decades of experience in seeking cures and treatments for devastating heart diseases, especially cardiomyopathies, and also happens to have the title of being the first physician ever to treat a Danon disease patient with gene therapy. And that will always be part of your legacy, along with the amazing work you've done at UCSD. So thank you for being here. And Dr. Joe Rossano is at CHOP, Children's Hospital of Philadelphia. And he is a pediatric cardiologist by training and in action, and has been working with us together for a couple of years. A great sort of trailblazer in the stars of cardiology today at ACC and AHA, and his work is very, very well known. And now has the title of having treated the first pediatric Danon patient with a gene therapy approach. So we're very happy to have all of you on stage, and I have some questions for you. Others may have questions for you, and maybe you'll have some questions for us. We'll keep it fun, right? So I'm going to start with Matt. Tell us about the experience of Danon for female patients with Danon, because the male experience is reasonably well known to this group. This is what we work on. Tell us about the female experience. And by the way, we haven't previewed any of these questions, but I think you'll handle them fine. Be easy.
Well, yes, so Dr. Danon's original paper did not include any information about females with Danon disease. The first patient I actually met was a woman with Danon disease and part of an extended family. And we realized pretty early on that the disease was probably going to be different in women and girls. There's somewhat of a fairly common story for the boys and men of intellectual problems, some mild muscle disease, and some cardiac disease. But the story for girls and women is really much broader. Some have stories similar to the boys, but some have very different stories. The original family that we are still in connection with, the woman that we first met is in her 60s, has not yet had a transplant, and really doesn't have any sort of muscle disease. So we got interested in this one particular family and started studying not just the boys, but also the girls and women. And I would say right now that's a process we're still working on. I think that we can describe the boys pretty well. For the women, it's still a different story. We're still working on that, Dawn. So we appreciate the data that you've provided for some of that. Well, and so the binder that Barbara mentioned, that's the rare disease binder. And so in our genetics clinic, patients frequently come in with binders because they've had the experience that Barbara has, which is that their doctors don't listen to them or don't look things up. So that rare disease binder that she has, I'm going to want a copy of that at some point because that information is pretty valuable because it's sort of a story of your disease, your condition. And that's what we're trying to figure out for the women as well, is what's their story, because they all have a slightly different binder, and we don't know what's in it just yet.
Excellent. Eric, tell us about your first experience with the Danon patient and how you identified him as a Danon patient.
It's actually pretty relevant to today's conversation because I was taking care of a young gentleman that presented to us rather acutely and extremely ill. And we were working him up for heart transplantation. And he also had this severe skeletal myopathy, which became apparent. And in the process of taking care of him, we did a skeletal muscle biopsy, and he was diagnosed with Pompeis disease. So there's clearly a theme here of misdiagnosis, and it makes me think a lot. I think one of the key elements of rare disease medicine is you never see something if your eyes are closed. So I think if Dr. Danon had kept his eyes closed and didn't question the orthodox, and just say, oh yeah, another kid with Danon, we might be still there today. And so in this case, I think, I was working with a young fellow, Stacy Clegg, I like to give her credit, and the patient was diagnosed with Pompeis and was getting enzymatic therapy for Pompeis and was getting worse. And Stacy was fighting with the pathologist, and this is not Pompeis, because the patient was around 30. She said, this doesn't make sense. Pompeis patients are younger, and this patient had Wolff Parkinson White, and she pulled me aside, and I mean no insult by this, and she said, I think this patient has Danon, I think everyone's wrong. And she was supposed to be doing another project. I said, well, focus on your project. You got to, oh, no. This kid has a disease called Danon, and you need to pay attention. And I said, what's Danon disease? At the time, there wasn't a lot of awareness. I mean, there's awareness, but not a lot, and it's a rare disease. And so she taught me, and she said, what are the chances that he has Wolff Parkinson White, that he's 30, it doesn't make any sense. And she and I brought I think being a physician scientist, we're in a unique position where we have a lab, and I see people in clinics. So we brought his skin cells down to my lab, and we sequenced it for the gene LAMP2, and we proved that he had Danon. And then he passed, and then about 3 months later, I saw a second patient. What are the chances that had Danon? And at that point, now had dealt with 2 grieving families. I saw the impact of this disease, and that it was having on these families, that no one knew what it was. And we now, because of newer technology, both patients died, but we could grow both of their skin samples into heart cells into my lab. So we used the technology that was responsible for a Nobel Prize to convert skin cells into heart cells called iPS, pluripotent stem cell. And so even after the patient died, we could study the pathobiology of disease, get to the root cause of why this gene causes disease. And that was 11 years ago, or 12 years ago. And that was kind of started my passion for it.
It's an amazing story. So maybe I'll ask Joe. You've seen boys and girls grow up with Danon disease. What is a sort of, not typical, but how would you describe the experience of a boy who's diagnosed, and what is their path over the several years that you've seen patients with Danon disease without treatment?
Well, it can be, it can be a really devastating disease, unfortunately. I think in the years past, similar to Eric's story and story from others, it was a late diagnosis. So these were generally children that got referred to us with hypertrophic cardiomyopathy, which seemed very severe and out of proportion in the severity from what we would have expected. And oftentimes the diagnosis of Danon wasn't made until very late in their disease, when they really were very end stage and either succumbed to their illness or underwent transplant. I think there is a better awareness today, and so people are diagnosed earlier, which is definitely better. It's really great if we have an effective therapy, which hopefully we do. But part of the burden then of knowing, one has a disease is then knowing what the future may entail. And that can be a very difficult thing for, the children as they mature and they, understand that more. And certainly from the parent standpoint of looking ahead and seeing what's the future going to be for my son or my daughter. Most of the time, they're really young children, affected our boys. So it can be very tough. Heart transplant is an effective therapy, and it can be very helpful, but we sure would like to avoid that if we could. frankly, that's what's so exciting about this. We don't really have a disease otherwise that alters the natural history, that really changes what is going to happen. So it's really a matter of surveillance and trying to intervene with a heart transplant before something really bad happens. So the prospect of having something that could cure the disease or greatly modify the outcome is really just tremendous.
For Barry, you've now treated a few patients and followed them over time using a gene therapy approach. What is your experience, and what are those patients' experience so far in your opinion? How have they done?
Well, we've actually treated 5 patients, and it's been a remarkable experience, both for myself, for the patients, and for the families, to have this opportunity on my part and on the patient's part to receive a therapy that will potentially really be effective in improving the clinical course of all of these patients. When we start doing studies like this, we don't have a historical perspective. And I remember when Eric, who's a colleague and friend, came over to me, he said, gee, Barry, I've got a great opportunity for you, which with Eric, I should have known to be aware at that point. He said, and I knew the work that he had been doing, and he said that he had now contracted with a company, which was Rocket, and put together a program, and they were ready to begin administering gene therapy to these young boys with Danon. Would I want to be involved? And I had had some experience with gene therapy in the past in my lab and in the clinic, and in doing clinical trials, and I thought this would be a wonderful opportunity. But I didn't know anything about Danon at that time. So I did a crash course and learned about it and said, this really is going to be meaningful. And then I met the first patient with Danon, and it really hit home how important this was to meet the mom and to meet the boy and to say that we had something that was going to potentially help him over the course of his life. And to be able to do that now 5 times, to meet 5 boys and 5 moms, has been an extraordinary experience for myself.
That's great. That's great. Do you have, maybe the cardiologists and internists and research scientists, do you have questions for Dr. Danon? If you were to meet Dr. Danon on stage one day, randomly in Times Square, what would you ask him? Any question they have for you, these gentlemen right here.
Well, yeah.
Yeah.
I mean, I think I would obviously express tremendous gratefulness. But what struck me today, and we see this all the time doing investigation, is you get the wrong result on experiment. And 99% of the time people say, oh, that was a fluke or someone messed up, and they leave it be. But we know that's where the science, the exciting science is from the quote unquote mistakes that most people pass on and say, oh, we can think of a lot of therapies now that have come, or diagnoses that have come from when most people ignore it. So what was the spark in you to not give up on this case and to chase it down and to define it, and to disagree with what I'm sure most people said, just leave this alone, it's Pompeis disease, what are you doing? That's my guess.
Well, it was fascinating. A disease that looks very much like Pompeis disease, textbook, turned out to be an enigma. Enigma is fun when you're doing this because then you can diagnose new diseases, and this was what happened at the end, and the cases start accumulating from all over the world, especially coming from Japan, with this not only histochemical electron microscopic findings, but the clinical triad. And also, apparently, we did not examine our patients' eyes. Apparently, they also have retinitis pigmentosa. And I wonder how many cases have retinitis pigmentosa, because I don't know the answer to that. But I think that's a new twist that, in addition to the triad, that they also have eye findings, and we don't know the reason why.
I've got a question for Dr. Danon, and I'm sure I'm going to share an experience that my other panelists have had. We've all had our manuscripts rejected by medical journals, and I've heard that your manuscript was initially rejected. What did you think, and what did you do?
Well, I didn't have a name. I guess that's what it was. And I sent an abstract of the paper to be presented in the American Academy of Neurology, and was just rejected. And I put an abstract to a neuromuscular disease symposium in the 80s in Montreal, and it was accepted as a poster, but not as platform, so they didn't think it was good enough to be platform presentation. But we did publish it at the end, and I think it was a big hoopla.
Dr. Danon, I would be interested, what advice would you have for young physician scientists? What advice would you have for young physician scientists starting on their career today?
What do I want to do?
What advice for young physician scientists?
Yeah. Well, the best thing is neuromuscular pathology. And the interesting part of neuromuscular pathology is that it's not billable, or the billing part is not that high. So there are not very many people being trained or inexistent in neuromuscular pathology. We're becoming like dinosaurs and probably going to be extinct soon. So, but neuromuscular pathology, once you fall in love with the microscope, you become an entirely different physician.
So I wasn't going to ask a question, but I got to follow up on that. So my dad's a pathologist. He's retired, so he's gone the way of the dinosaurs a little bit. And maybe you don't know this, but we actually don't have medical students use microscopes very much anymore. That is sort of a thing of the past. It's archaic. You're dating yourself by talking about microscopes. So I'm curious, do you have a sense of what's going to happen next to neuromuscular pathology? Because we don't use microscopes very much. We do not do muscle biopsies anywhere near as much as we used to. What's going to happen next, do you think? Because we're going to lose this art.
Well, in the old days, the people who did neuromuscular pathology were actually clinicians. And a pathologist, a general pathologist or subspecialty pathologist felt very threatened by these guys coming in and doing microscopic work when they didn't do any pathology. So what happened is the pathology boards got into it, and so the clinically trained neuromuscular pathologists started becoming non-extinct. The same thing happened with basic brain neuropathology, and most primary old neuropathologists were neurologists who also did neuropathology, and they knew how to cut brains and stain them and make fantastic discoveries. By then, the board of pathology took over neuropathology. Now there are very rare people training in neuropathology after they finish their pathology residency. So that's another twist in the field.
Question for Joe, actually. Do you think that increased and routine genetic testing or screening in patients with heart disease may uncover more Danon cases and other similar genetic diseases? Do you think that's the future?
I think so, and I suspect, I'm interested in the panel's opinion on this, but we likely identify disproportionately the severe cases, and that there's likely more of a spectrum that we're just not picking up on, and that could be very informative for future therapies as well. That was certainly the case with hypertrophic cardiomyopathy, the initial studies, devastating, horrible disease. Now we know large spectrum, many people with mild and moderate disease do very well. So I think with we do a lot more genetic testing now. We certainly try to do testing in all of our patients. And so I think we're likely to pick up on, a greater spectrum of disease, which I think will be informative for the field.
Eric, what is your appetite for pursuing other similar diseases in the future, like Danon?
Yeah, we talk about this a lot in my lab and in my clinical practice, and Barry and I talk about it. And I think it's really relevant to Rare Disease Day in general. But there's a term you might hear, doctors say, which is idiopathic. And my daughter heard me saying it, and she says, "Did you say idiopathic?" And I thought about it. I said, maybe that's a better term, because idiopathic is one of these fancy terms that doctors use to hide the fact from patients that we don't know what we're talking about. We say, "Oh, it's idiopathic." I say, "Oh, idiopathic." In reality, it means we don't know. And there's a whole field of cardiology, idiopathic cardiomyopathy, and a whole class of patients far beyond Danon that actually have a root cause of their disease. And I think, and it's because of gene therapy and a lot of new technology, whether it's inhibiting RNA, whether it's treating, directly delivering our gene therapy without a virus, whether it's changing your RNA with these new technologies, we're going to be able to go after every single one of them. So we'd like to see a day, and especially now that genetic testing has gotten cheaper, and where we can eliminate these diseases. It's just going to take, for too long we've been trying to take this all-encompassing approach. So I give a lot of credit to companies that are focusing on rare disease, because forever it's been like, let's come up with a blockbuster and treat everybody and we'll cure heart failures, and but the reality is biology is not so simple, and that we're going to have to develop tailored therapies for each one of these diseases. And Danon, I'm hoping, is a paradigm for many, many others. It's the tip of the spear, is how I look at what we're doing for many other diseases, is my ultimate hope.
Yeah, that's really an important point, Eric. I think we as physicians have been lumpers for many years. And we were lumpers not because we wanted to be lumpers, but because the tools we had available allowed us only to be lumpers. But there's been a transformation in science and in the way we look at human disease now that's going to allow us to be able to really understand diseases from their very basic genetic cause and treat it in that way. And I think that we're on the verge of a revolution now in the way we manage disease in patients. And this is a beginning of that.
So I have to say that in the history of the universe, this is a special moment, right? It's almost like the Big Bang happened to lead to a few special moments, and this is one of them, where Dr. Danon is together with the treating scientists and physicians, together with many of us who are working behind the scenes to find cures for Danon disease, right? This is uncommon. It doesn't happen every day. So we have about 7 or 8 minutes left. I'm going to take questions from the audience and then ask the panelists for one final message or thought to put out into the universe. This one time I'm previewing the questions so you have time to prepare. But meanwhile, any questions from anyone? And I think we have a floating mic as well from Kevin. Oh, there, great. And please actually introduce yourself when you're answering the question, if you don't mind.
Hi, this is Jonathan, Rocket's Chief Medical Officer, and this is a question for Dr. Danon. So some 19 or 20 years transpired between your original report and then the subsequent publication where the LAMP2 gene was identified as the causative issue behind Danon disease. At what point during those 19 years did you think there could be a single gene that's responsible for all of these heart and neurologic and muscular issues? Was it soon after? Was it not until the gene was discovered and published? At what point did you think there might be a single molecular cause?
In the 19 years from 1981 to 2000, between these two papers, did you ever predict that one gene would tie it all together?
The answer is no. I had no clue. I had no clue. The answer is not. But to follow the question of idiopathic, I described a disease in the 1990s called critical illness myopathy. And this was patients who were paralyzed with myoneural junction drugs and who were giving high doses of steroids, which happens in the VA, in the ICU setup. And in the 7 days, something rather biblical, they become totally quadriplegic. And this was thought it was some sort of a neuropathy until I did a biopsy and showed it was a very specific primary myopathy, and electron microscopy showed myosin deficiency, certain part of the protein. And this is from the bed to the bench. At that point, I took the muscles, I went to the anatomy lab, and they were doing molecular biology on various proteins, and this turned out to be acute steroid myopathy, which destroyed only the heavy chain myosin and muscle. So this was an idiopathic disease, supposedly, which turned out to be, if you follow it and you fish for it, you end up with a new disease and with a cause and probably treatment. So this is to follow the idiopathic comment, which is so apropos to this chat.
Over here, Kevin.
Hi, my name is Anirudh Koneru. I'm the CFO at Shaping Foundations, and I had a question in general for like any of you. There's a saying in medicine, like when you hear hoofbeats, think horses, not zebras. And going back to the point that you guys made about like physicians becoming almost sort of lumpers, like trying to come up with a cure-all, like coming up with a common diagnosis for as many like general symptoms as possible. Do you think that that's going to change in the future, like the idea of like rare diseases becoming more focused on, I guess, considering that genetic research is becoming more prominent? Do you think that that's something that's in the future for medicine?
I think that, yeah, that's a great question. And I think that not only are rare diseases going to become more common as we discover genetic basis and do genetic testing, but people with common diseases are going to be subdivided into different causes, which are going, and that's going to be very helpful because they're going to respond to different therapies in different ways. So I do believe that's very much the way of the future.
It's a great question, by the way.
If I can add on to that, I'd say that the good and the bad of it is that when I went to medical school, well, you're pretty young, but when I went to medical school, we didn't learn about any of this sort of stuff because rare diseases, you couldn't diagnose them. You get some strange pathology, neurology people diagnose them in weird circumstances. It just wasn't something you had to worry about as a physician, so we didn't learn about this stuff. So we missed all these rare diseases. The tools that we have now, the genetic testing that Joe was talking about, that means we have to sort of pay attention now. So we're going to have many more genetic diagnoses just because there's genetic testing. I mean, raise your hand if you've had your DNA tested by, say, 23andMe or Ancestry.com, right? So, you know, there's like 20% of the room, 10% of the room has already done that potentially. That's going to become very common. And so we'll be doing genetic testing on all of our medical students when they enter medical school So they can learn what their conditions are right off the bat, so to speak, or their risks. And that's really going to help us make these diagnoses. And the common disease as well, Barry's point's a very smart one. I mean, heart failure that is treated as heart failure is going to be divided into these small categories based on these new markers. So it's going to be transformative.
Hi, I'm Frank Ferreira, but my question is just a transcript from online. So this question comes from Vardika Mishra. It's a question for clinicians. How can we make sure that when the patients first present themselves with minor symptoms, how do we make sure that the doctors seeing them for the first time have better training or guidelines for diagnosis? What can be done to make the path from symptom presentation to final diagnosis shorter?
So we're hoping, it's funny, I'm going to look at the poll over here because I don't know where the voice came from. There in the back.
He's the MC for the day. He's in the corner.
There we go. I just couldn't see you in the back there. So it's a challenge. We know for many rare diseases that it takes 5 to 10 years to get diagnosed. So that's a long time to sort of have symptoms. And obviously really minor symptoms are hard to have your physicians pay attention to. So we don't have a perfect answer for that. From the genetic standpoint, the fact that genetic testing is more and more accessible and it's going to happen to more and more individuals, that's going to help drive this quite a bit. It will create another potential problem. So what do you do if your 3-year-old gets genetic testing and is diagnosed with something they may not be at risk for 4 or 5 decades? So it'll create some new challenges, but we hope that genetic testing will be part of this. And then organizations like NORD, which is the National Organization for Rare Diseases, they're promoting right now rare diseases. I think there's 30 or so centers around the US that are now NORD Centers of Excellence. And so we're going to elevate the idea that rare diseases are out there. Actually, they're quite common when you add them all together. And that will hopefully get our clinicians to be better at making these diagnoses. But it's going to be a challenge.
So thank you for your time to educate us on Danon disease and promoting the awareness. So I think Dr. Greenberg talked about treating female patients versus male patients, and then you're talking about dialyzing earlier. So what are the differences in experience in treating pediatric patients versus adolescents, older kids?
The difference in treating pediatric patients versus older patients, adults.
Well, I think that number one, there's a greater understanding on the part of adult patients. I don't treat very many children, and but I found that in treating the patients with Danon that I've had who have aged between 15 and 20 years, it's quite different in dealing with them. There's much greater involvement of their parents in that. And I think that, to me, as a clinician, was something that I really wasn't as used to in the past. And in treating Danon, and I think my colleagues can comment on this, you really don't treat a patient, you treat a family.
Well said. Well said.
Hi, Spencer. So I was wondering, how would you say you see the future of science going forward, working in tandem with the supply chain for this sort of thing? Because these are very rare diseases, and finding treatments after finding a cure must needs be much more difficult as we start looking for more specifics. So could you speak to that?
Well, yeah, I think that's a very insightful question, and you know, it gets to the root of, pardon me, what is translational medicine? And you know, if you think about how therapies are developed, it used to be considered very successful if you could develop a therapy in 20 years or 10 years from inception, and we can't tolerate that anymore. So we have to think about how we bring costs down, as you mentioned, supply chain, how we improve the relationship in the regulatory process to to not be as burdensome, or I mean, I think burdensome is a strong word, but to be as efficient as possible. Knowing that maybe for a scientist or 5 years doesn't seem like a long time, but for a patient, 5 years is an eternity. So I agree with you, but we're going to have to work in tandem, and I think that's happening already. I think, people say to me, "Oh wow, this has been so fast with Danon." Yes, but it's really 11 years since, and even 6 years since first started developing the gene therapy. So I think we can do better, and I think we will as technology improves. But it's a fundamental question, and if we don't take that question on, we won't make the progress we want to.
So we'll wrap up with one final question. And I've lived in New York for a long time, and I was told that if you make a wish and put it out into the universe, it comes back. Could take many years. As long as you're west of 7th Avenue. And we are technically just west of 7th Avenue. I've tried this, it works. What is your wish to the universe regarding Danon disease?
I mean, I think for me, I think it's hopefully it's obvious. We want to eradicate it. We want to obliterate it. That's our goal, and it's not just a wish; it's a mandate for me. So I think sometimes wish has a connotation that it's not something that's going to happen or it's pie in the sky. But this is a singular focus right now. That's we can do it. So we have to do it. If we can, I mean, you met with these families, you saw Bar, then we are mandated, and so I think, but no one can do it alone, but together we can do something beautiful.
Yeah, that's well said. I'd wish for a cure. I mean, I think we want to cure this disease, and I mean, Eric put it right. We have the ability to do that, and we should do it. And it's an exciting time. I think it is possible. It is not a pipe dream.
I don't think I can really do much better than that. I guess I'll wish for a trial in women at some point. That it's a shout out to all the X chromosomes out there in the audience. But yeah,
it's in the works. Where's Paul? When are we starting? Seriously, we're planning to start in the next year or two. So yes.
I guess I think of one word, and that is awareness. And I'm hoping and I'm wishing that this trial will raise the awareness about not only Danon, but other rare disease. And also, since it's the first gene therapy of a monogenetic disorder, the success of this program translates to other programs through that awareness.
Beautifully said. And the last question for you to close it out, Dr. Danon. What is your wish?
Well, a lot of young neuromuscular scientists who had great promise in the field of neuromuscular disease switched to Alzheimer's because that's where the money was. And there was a common disease. Muscles hides a lot of mysteries and a lot of diseases in them, both yet to be discovered or to be treated with various either immunological or genetic type of treatments. And I think it's a very exciting part of the body.
It's an honor to have all of you here. Thank you for coming, and more to come. Stay tuned. Thank you.